I will be wearing a mask to give this presentation since I'm just recovering from COVID. But now I'm negative and safe, but just occasionally coughing. So yeah, let me see my face. Hello, I'm Shuchen. It's an honor for me to be here to present our current ongoing project. We call that the GET-nQTL, the embedding transformed centrality-based network QTL analysis. And let me show you some brief introduction here. So in the past few years, the single-cell RNA-seq from multiple individuals across the different cell types and different experimental conditions offer great opportunities in the genomics research. We can look at the gene expression at the cell level resolution and try to analyze some personalized analysis. So this data enable us to do the estimation of the subject-specific gene co-expression networks. For example, we have the single-cell gene expression data from different individuals. We can estimate the pairwise gene correlations and thus construct gene co-expression networks. And why we're caring about this is because that some genetic variants linked to the disease might not be associated with changes only in the gene expression, but some of them are associated with the gene co-expression network. For example, in the normal tissue here the TF may regulate the expression of the gene A positively. You may see a positive correlation between them. But if some mutations or some variants happen in this TF, and it may lose the control of the expression of gene A and even prevent it from expressing. So you may see, although you may or may not observe the difference in the expression level things, but if you put them together to see their co-expression behavior, they may be different. That's the point here. So given the personalized code expression networks estimated from the single-cell RNA-seq data and together with their genotype information, we can ask whether the properties of this network vary with the genotypes. That leads to the idea of the network QTL. That's different from the eQTL, which focus more on the expression level, and the network QTL, we focus more on the co-expression network level. And in this project, we are trying to develop a novel network QTL approach to identify the effect of genetic variants on the co-expression network properties. So the overall idea is trying to find the association between some network feature and the genotype. So we start with looking at the centrality, the centrality of a node of the gene in the network. It can be the degree of betweenness or page rank, something like this. And we did some experiment to see, okay, we need some signals here. On the right-hand side, you will see in some case, the degree may be different from the different genotype, but sometimes they may not have significant difference. And after looking at that, we are wondering whether, okay, so all the information we have here is from the data set we are working on. We estimate the co-expression network from. Whether we can bring more information from the existing knowledge. So we're looking at some foundation models with the ability to generate the gene embeddings to combine the information from the specific data set and the existing knowledge together to do the analysis. So that leads to the idea of the GET-nQTL. We want to find a way to quantify the gene role in the network. We'll call that GET centrality later, and detect how these gene roles are associated with the genotypes. So the idea is like that the role of a gene in a network can be described by its neighbors. Another is that, okay, how to quantify the importance of these neighbors? We can borrow some information from the existing foundation models, large language models, and can embed the gene information that we already know into quantitative gene embeddings. And we can combine the correlation from the network and the gene embedding together to evaluate the change of the gene role in the network. It will be a 5-step here. And first, we need to estimate the gene co-expression network. Here we use the DOSER, a debiased personalized gene co-expression network estimation algorithm. And it can provide a metric to quantify the genes measured with the high noise, which can do better. Let us do better in the co-expression network estimation. And after we get co-expression network, we try to find some neighbors with higher absolute correlation. For this case, the neighbors may be in A and C and F, this node. And after we get the correlation information from the network, from the data we have, we can try to find some additional information source. Like in the last year or this year, there are a lot of foundation models in the single cell data area, GET-nQTL, TPT, something like this. And we try to start with GPT, since its idea is quite straightforward and very easy to use. So the idea is that we all have experience talking with, I guess most of us have experience with ChatGPT. You can ask ChatGPT whether it knows some information of the gene, and it will give you a brief introduction to the gene, which indicates that this LLM, they were trained with some biological data, so they have the ability to generate quantitative embeddings to capture some underlying biology. So what you need to do with GPT is that you prepare a short text description of the gene you want, like its name, its ID, its type, and a short summary. You put this text to the GPT model, and they will give you embedding for that. And there has been shown some successful applications in the downstream analysis in this GPT papers that these embeddings can help us to capture some information for like gene functionalities or cell annotation. Yeah. And we are going to start with GPT as the source of the gene embedding. So now we get the two information source. How do we combine them together? Well, very naive idea is to, okay, we just, although there are a lot of details in applying this step, but the overall idea is like, okay, we have the embedding here, and we will take the weighted average of the neighbors' embeddings weighted by their normalized correlation strength with the central gene to get we call it the GET centrality of the central gene. But it will represent the role of this gene in this network. And we have found some evidence in the real data that this approach can overcome some false negative if we only look at correlation and centrality themselves. Maybe not enough time for me to show. The last step is we try to implement several statistical hypothesis testing to run this kind of regression with the covariates we may want to see what is the statistical significance in it. So let me show you some results. We are working on more results, and this is a data set that we already have some result on it. So this data set will have 119 healthy individuals, and working on the CD4 plus T cells, and they also have a time point of cell activation, the 4 time points, and 2 major cell types. That could be provide us with a different cell context. So first, the GET centrality do review NQTL, NetQTL genes. So in this UMAP, each point represents its gene centrality, GET centrality in one subject gene co-expression network. So each point is a subject. And colored by their genotype of the donor for the certain SNP. Here we pick one gene and one SNP, and you may see there is an overall very significant difference between the two genotypes colored by red and blue. And also this effect is significant if we look at the 8 different cell contexts, like different cell type at a different time point after activation. So that's the first result. And the second part of the result is that this GET-nQTL effect varies in different cell contexts. So if we look at here, focus on the right one, at the zero hour, at the resting state of the cells, we do not observe a significant difference across the genotypes of this GET centralities. But with the time increase, you see that the 5 days cells, we can observe there a very clear difference across different genotypes, like the red, the green, the blue one, they separate. And the third one is, as we mentioned, some of the nQTLs, they detected as the eQTLs, means that they also have the different expression level. But we also observed that some of the nQTLs, they are not detected as eQTLs. It means that do not have a significant difference in the expression level. Well, for example, here, this SNP and this gene SNP pair was not detected as an eQTL in the analysis from the data from the paper from which we use its data set. But it do have NQTL significance here. So to conclude, in this project, we try to combine the gene network information estimated from the specific data we have and the existing biological information from the labs we already have to put them together to improve the detection capabilities. And the challenges we're facing are like we have a large amount of multidimensional multiple hypothesis testing problem. And we have found several, we are trying to implement various statistical testing to fit different data needs. This is still an ongoing work, and we are trying to make this GET-nQTL flexible pipeline for the network QTL analysis, also including the calibration, the p-values, validation on the different data set, and also investigation of what exactly these gene embeddings can provide with that. Okay, so let's thank my advisors, Professor Sinduskas, and also my team member, Zhongxuan, for their contribution in this project. Thank you for listening.
Time for a quick question, or people are ready for lunch? Question? Oh, there is a question. Okay.
Hi, great talk. I had a quick question. So have you actually looked at the overlap between your GET and QTLs and what you would get if you just correlated or if you just tested for association between SNPs and doser centrality, doser estimated centrality?
Oh, that's one I mentioned, but I don't have enough time to elaborate on that. It's like we have found some evidence in the real data that if you only look at some centrality, then the centrality value may not change, but the gene's neighbors it may change. It looks like you have the same, if we only look at the list of the correlations, the gene correlation with others, you may not observe a significant difference, but actually the genes they link to, this correlation link to, they change. So by using GET-nQTL, we can detect this false negative cases.
Right, yeah, it would just be interesting to see how different those two are. It would just be interesting to see, like, globally how different those two are.
Different.
Yeah, we can just chat offline.
Oh,
Thanks.
Oh, maybe later. Thank you.